Good morning, students. You're welcome to your third lesson on Biology of Heredity 1. In today's topic, we're going to be treating patterns of transmission of traits. And our subtopic will be on dihybrid crossing. Dihybrid crossing. In our previous lesson, we discussed monohybrid crossing, single factor inheritance. In this one, we are going to be treating the transmission of two traits simultaneously. And we are going to see how it is being transferred, how the law of Mendel, that is second Mendelian law, applies to this, the law of independent assortment of genes. Is going to reflect in this dihybrid crossing we're going to do, and I want you to be handy with your writing materials, as we're going to be doing a lot of illustrative work. So, if you're ready, let's move on to the first step of our discussion. All right, now, in the first step, we are going to ascertain the two pairs of genotypes of the parent generation. In other words, the ones we are considering, we're going to be considering two traits. For today, we're going to be considering height and complexion. And we're going to use two individuals, of course, married couples. And we're going to use them to do this example. That of the father's genotypes for the two traits are going to be heterozygous dominant tall gene, as you can see on your screen, heterozygous dominant tall gene, and recessive short gene. That is for the height. Then for his complexion, we're going to have homozygous dominant dark gene and recessive fair complexion gene, all combined together. And because they are two traits, that's why we call it dihybrid. Then for the mother's genotype, we're going to be seeing almost, of course, the same thing as the father's genotype. You have heterozygous dominant tall gene with a recessive short gene, and also heterozygous dominant dark gene with a recessive fair gene. So your first step in dihybrid crossing is to ascertain the genotypes of the parent generation, as you can see. Then the next step is to find the possible gene combinations. Find the possible gene combinations. And I'm going to show you how to do your finding of the possible gene combinations as you watch on your screen. As you watch on your screen. The possible gene combinations. Okay? Now, the first thing you do is to write out the genotype of the parent's generation as I've written them. Now, what it means is, in how many ways can these two traits in the father be assorted? In how many ways can they be differentiated or assorted into different gametes? That is what it means for possible gene combinations. And we're going to use our monohybrid crossing pattern to form these possible gene combinations. Of course, if you do it, you're going to be seeing patterns like this. In the first gamete, you can see the dominant tall gene coming into the same gamete with the dominant dark gene complexion. In another gamete of this same man, you can have, of course, in the male, and every organism that is involved in sexual reproduction procedures, the male always produces millions of spermatozoans. And in those millions of spermatozoans, there are different gene combinations. That is exactly what you're seeing now. So in the next gamete, you can either see a dominant tall gene forming with a recessive fair connection gene as in the next gamete. And then you can also have this possible gene combination as well, where you have dominant dark gene coming into effect with recessive short gene. This is another possible gamete that can be formed in such a man. All these things are among the millions of his spermatozoans. Then you can also have this as well. You can have a gamete of his where you have the recessive short gene with the recessive fair gene in another gamete of the same man. It's quite interesting, the study of genetics. So this is the possible gene combination that you can find in the man. Now, because the man has the same genotype with the wife, there is no need for us to go into its assortment or the way they are going to combine, because you will still get the same result. I will leave that of the mother's genotype for you to do. Do it on your own. You will still get the same result. Let's go to the next one. Let's go to the next one. The next one is to find, of course, we've done that one. We did it in the previous one. Find the possible gene combinations. We are done with that one. The next one is to draw your Punnett square. And we're going to draw the Punnett square immediately. First of all, you make your vertical line. Your vertical line intersecting with your horizontal line in this format. And then you make 4 columns on the vertical dimension. 4 columns on the vertical dimension. Also, you do 4 columns on the horizontal dimension. Okay, now the man's possible gene combinations are going to occupy the 4 columns on the horizontal dimension in this form. This is the symbol for the male gamete. It's going to be combining. You insert the possible gene or possible genotypes for the man's gamete inside the horizontal column on top, just as I'm showing you. The next one, then you have this, and then you have this. So those possible gene combinations you got, you insert them for the man there. Then for the woman's zone, it comes in the vertical line, where you have the same possible gene combination in her eggs. Then you draw for this one, you have it as well. And then the last one. The last one, you have it in this format. All right. So you have drawn your Punnett square and you've inserted the possible gene combinations. All right. Now, your next step, which the next slide will show, will ask you to match these possible gene combinations to form the offspring. Inside these boxes, you're going to see how this gametes are going to be crossed or matched to form the offsprings in the box. And immediately I'll go into that. Let's see the possible offspring formation. In this first box, you're going to see that this gamete from the man and this gamete from the woman, they are going to form this kind of offspring with genotype homozygous dominant tall and homozygous dominant dark genes. That is for the first offspring. The second offspring, you can now connect this first gamete of the woman, you connect it with this second gamete of the man, you'll get the next offspring, which will give you, I want you to sketch it for yourself, which will give you dominant tall gene with dominant dark gene, and then a dominant tall and recessive. You're going to have, sorry, go back to it and make the correction. You're going to have homozygous dominant tall genes for this particular child with heterozygous dominant dark and fair complexion gene. That is for this box. Now I want you to do for the rest of the boxes by crossing them accordingly. Okay. Now, if you can do for the rest of the box, remember you can do for this particular gamete of the woman, crossing it with this the other gamete of the man, and you will get the result of the offspring. Follow the same pattern and fill up the whole boxes, because my screen cannot contain all of this, but you can do it in your own paper. Please fill up all the boxes. Now let me give you another example. For this particular one of the woman's gamete, it will combine here, where you have dominant tall gene with recessive gene, and then you have homozygous dominant dark gene. I have done another example for you in the box. You combine this one and this one in this box. That's how you combine it. Everything in this line must align with this first. You go to the next column, you do it. The next column, you do it. When you finish matching it, then you do the following things as your assignment. When you finish matching all the boxes, write down the genotypic ratios and the phenotypic ratios of all the offspring. Your assignment, write down the genotypic ratios and all the phenotypic ratios of all the offspring. Thank you until we meet again in our next class.